Imagine you're designing the ultimate defense system for a brand new, highly vulnerable human being, a newborn. You wouldn't just give them bricks and mortar, right?
I mean, you'd want blueprints.
Exactly. You'd give them a programming manual, like the software code to tell those defenses exactly how to build themselves, where to go, and how to fight. But for decades, when we've thought about early infant nutrition, whether that's breast milk or formula, we've mostly just thought about it as those bricks and mortar.
Right. We just count the calories, the broad fats, the carbs, the proteins.
Yeah, assuming it's just the raw fuel needed to make a baby grow bigger.
We basically view it as a bulk delivery of construction materials. The idea was always, just pour the energy in and the baby's internal machinery it'll figure out the rest.
But then you look at the immunology of a newborn and suddenly that whole construction material metaphor just completely falls apart. So, welcome to the Deep Dive. Our mission today for you listening is to explore a truly mind-blowing 2026 paper from the Journal of Science.
Yeah, by Fan and colleagues.
Right. And it fundamentally challenges how we view early life nutrition. We are going to unpack how a single, highly specific molecule in a mother's diet can literally reprogram a baby's immune system for the rest of their life.
Okay, let's unpack this.
Yeah.
Because it is wildly fascinating to think that food isn't just fuel, it's biological code.
It really is the absolute definition of a biological software update, and this paper, it completely changes the paradigm. While we typically view nutrition in those, broad macro categories we mentioned, this study zooms in on a surgical strike.
A very precise stroke.
Exactly. It draws on a highly rigorous immunological study detailing experiments on both mice and human preterm infants, isolating one specific dietary molecule that independently instructs immune development.
Right, but before we get into the lab results, we really have to understand the source of this mystery. Because human breast milk it's famously complex.
Oh, incredibly complex. It's packed with antibodies, cytokines, different proteins, and a whole microbiome of its own. Historically, science has just attributed the immune benefits of breast milk to this whole package working together in synergy, like a symphony where you can't really isolate a single instrument, right?
Yeah, exactly. It's been viewed as this intricate sort of chaotic symphony, which actually makes it notoriously difficult to study mechanistically.
Because there's so much going on.
Right. If a baby gets healthier, was it the antibodies? Was it the probiotics? The proteins?
Yes.
But these researchers asked a really radical question. They asked, could a single dietary molecule in that breast milk be doing the heavy lifting for long-term immune development all on its own?
And the molecule they put in the spotlight is called trans-vaccenic acid, or TVA, which brings up a massive question for you listening right now. Wait, a trans fat? Aren't we told by literally every doctor nutritionist on Earth to avoid trans fats at all costs?
Yes.
Are we seriously saying a trans fat is the secret software update for a baby's immune system?
I know it sounds totally counterintuitive, but it's a vital distinction we have to make here. We have to clearly differentiate between harmful, industrially produced trans fats.
Like the ones in processed food.
Right, like oleic acid, which is found in partially hydrogenated oils. Those are artificial and absolutely linked to cardiovascular disease. TVA, on the other hand, is a naturally occurring trans fatty acid found exclusively in ruminant products.
So we're talking about beef, lamb, and dairy.
Precisely. And it is the most predominant natural trans fat enriched in human breast milk. They behave very differently in the body. In fact, epidemiological studies suggest circulating TVA is actually associated with lower systemic inflammation and lower adiposity or fat storage.
And here is the kicker. The human body cannot synthesize TVA on its own. It comes 100% from the maternal diet. But hold on, if TVA is found exclusively in beef, lamb, and dairy, and the human body can't make it, what does this mean for, a vegan or vegetarian mother? Is her baby just missing out on this software update entirely?
That is a fantastic question, and it's a crucial real-world implication of this research. The short answer is, well, human biology is incredibly resilient.
Okay, good to know.
Right. There are likely other undiscovered dietary molecules or compensatory pathways that help develop the immune system in babies of plant-based mothers. The immune system has redundancies. But what this study highlights is a specific, highly potent evolutionary pathway that we just didn't know existed.
And it completely changes how we might think about optimizing infant formula, right? Which currently contains, I'm assuming, absolutely zero TVA.
That's right, zero.
Okay, let's do a quick checkpoint for you listening to make sure we've locked this in before moving on. We've got a lot of density today, so we'll do these little mini recaps.
Good idea.
So bullet 1, Breast milk is complex, but researchers isolated one specific fat, TVA. Bullet 2, it comes only from the mother eating dairy or meat. Bullet 3, this is a good natural trans fat, not the bad industrial kind.
Perfect summary.
But to prove that TVA isn't just a passive ingredient, but an active biological instruction, the researchers had to isolate it. And to do that, they turned to mice.
Right, because biology is messy, so you need a heavily controlled environment. They took lactating female mice and fed them either a standard control diet or a diet enriched with TVA. Specifically, they used either 0.1% or 1% TVA.
Okay, a very controlled amount.
Exactly. Crucially, the pups only received this TVA through nursing, so exclusively through the breast milk.
And the physical results in these pups were wild. I mean, they didn't just get fatter. In fact, their overall body weights were completely normal compared to the control group. So it wasn't just extra calories acting as bulk fuel, but inside. The pups drinking the TVA milk had significantly enlarged spleens and thymi at 2 to 3 weeks of age.
And just for context for the listener, the spleen and the thymus aren't just random organs. They are the primary command centers and training grounds for the immune system.
Exactly. So they looked closer at the thymus and spleen at a cellular level, and they saw a massive expansion of what are called naive CD4 positive T cells.
Right, naive CD4 positive T cells.
And for a memory trigger, I like to think of these naive cells as a standing army of raw, unassigned recruits. They're the blank slates of the adaptive immune system. They haven't been programmed to fight a specific virus yet. They're just sitting in the barracks waiting for an assignment. And the TVA diet was basically recruiting a massive army of them.
But having a big army doesn't really matter if they're untrained or exhausted. What the researchers needed to know was how ready these cells were. So they used advanced genetic sequencing, single nuclei multiomics.
Wow, okay.
It's advanced stuff. They used it to actually look at the physical 3D shape of the DNA inside these cells. And what's fascinating here is they found that these naive T cells weren't just numerous. Their genes were physically primed for action, ready to deploy to the lymph nodes without being prematurely exhausted.
Now, whenever we talk about diet and immunity, I feel like everyone immediately points the finger at the gut microbiome, right?
Oh, always.
So did the TVA change the gut bacteria, which then somehow changed the immune system secondhand?
They checked exactly that. They used advanced ribosomal RNA sequencing, basically a genetic roll call of every bacteria in the gut. The technical term is LymeSec, but what it really does is let us map the entire microbial landscape.
Whoa, wait, LymeSec. Are we making margaritas or doing science here? What did the roll call actually show?
As refreshing as that sounds, the science showed something entirely unexpected. The TVA did not substantially change the microbial landscape.
Right.
The microbiome variations were based entirely on which litter the pups belonged to, essentially which cage they grew up in, not the TVA diet.
So they completely bypassed the gut microbiome. The TVA was acting directly on the immune cells themselves. It's wild to think that a literal fat molecule can bypass the gut bacteria and dictate gene expression in a totally different human being. That is epigenetic origami.
That's a great way to put it.
But that brings us to the most mind-bending part of this entire deep dive, the molecular domino effect, the how.
Yes. How does a lipid molecule floating in milk actually talk to the DNA coiled up deep inside an immune cell?
Right, because DNA is locked tight inside the nucleus of the cell. You'd need a highly specific chain reaction to get a message from the outside of the cell all the way into the vault.
And the pathway they discovered is remarkably elegant. Let's break this down step by step. It starts at the surface of the naive T cell with a receptor called GPR43.
Okay, GPR43, got it.
Think of GPR43 like a parking brake on the cell's immune development. Step 1. TVA binds to this GPR43 receptor and actually inactivates it.
So TVA isn't starting the engine, it's releasing the parking brake. And the moment that brake releases, the car can roll forward.
Exactly. By inactivating GPR43, step 2 occurs. It triggers an internal cellular signaling cascade. Specifically, it increases the production of a molecule called cyclic AMP, or CAMP, and activates an enzyme called pKa.
Okay, CMP and pKa.
If GPR43 was the parking brake, CMP and pKa are the cellular messengers who just took their foot off the clutch and are now sprinting toward the nucleus with new orders.
Okay, so TVA hits the receptor, releases the brake, the messengers sprint to the nucleus. What happens when they get inside the vault?
This is step 3, and it involves a protein called CTCF. CTCF is what we call a 3D genome organizer.
Okay.
Human and mouse DNA is incredibly long. I mean, if you stretch the DNA from just one microscopic cell out, It would be about 2 meters long. To fit inside a tiny nucleus, it has to be folded with extreme origami-like precision. CTCF is the origami master.
The origami master, I love that.
Right. When the Gampion PKA messengers arrive, they increase the activity of this origami master.
And the way the origami master folds the DNA completely changes which genes are accessible to be read by the cell, right? Like if a gene is folded on the inside of the shape, it's hidden. If it's folded on the outside, it gets activated.
Precisely. Which brings us to the final step, step 4. CTCF cooperates with a transcription factor called TBX21, also known as TBET.
Okay, PBET.
Together, they physically open up and read specific genes, like the one for interferon gamma. This pushes the entire immune system toward what we call a Th1 biased state.
Okay, Th1 biased state. We need to unpack this because understanding the difference between Th1 and Th2 is basically the whole key to why this matters for a newborn.
Right. In a typical newborn mouse, and to some extent human neonates too, the immune system is highly immature and leans heavily toward a Th2 state.
Th2.
Yeah. Th2 immunity is great for fighting parasites and dealing with allergies. You can think of Th2 like deploying a sticky net. It's helpful for certain things, but it's slow and not ideal for fighting off sudden, aggressive, acute infections.
Right. If someone breaks into your house, you don't want a sticky net, you want a SWAT team.
Exactly. T1 immunity is the SWAT team. It's specifically geared for fighting viruses and intracellular bacteria. It is highly aggressive and targeted. What the researchers proved is that TVA is physically reaching into the DNA and flipping the switch from a Th2 default to a Th1 viral fighting stance.
Okay, time for another quick recap for you listening because that was a very dense chain reaction. Let's lock it in with our memory triggers.
Let's do it.
Step 1. A mother eats dairy. The TVA fat goes into her breast milk. Step 2. The baby drinks it, and the TVA hits the baby's immune cells and releases the GPR43 parking brake. Step 3, that sends the CAMP messengers into the nucleus to tell the origami master, CTCF, to refold the DNA.
Right.
And finally, step 4, that new DNA shape builds a massive SWAT team of Th1 immune cells ready to fight viruses. It is just breathtaking that we've decoded this level of detail.
It really is. But having a primed immune SWAT team sitting in the barracks is useless if they don't know how to fight when a real war starts.
Right. We have this beautiful genetic origami happening in a sterile lab environment, but biology is messy. If this baby mouse is actually exposed to something nasty out in the real world, does this new Th1 army actually offer protection?
To find out, the researchers challenged these tiny 10-day-old mouse pups with 2 severe real-world pathogens. First, H1N1 influenza A, which is a severe respiratory virus, and second, Salmonella typhimurium, a severe bacterial infection.
Why both?
They wanted to test both a virus and a bacteria to see the breadth of the protection.
And the results weren't subtle. The pups that had been nursing on the TVA-enriched milk had significantly enhanced survival rates across the board. They had vastly lower pathogen loads, meaning when the scientists looked at their livers and spleens, there were physically fewer salmonella bacteria colonizing their organs.
Yeah, a massive reduction.
And when they looked at the lungs of the flu-infected pups, the TVA group had significantly reduced lung tissue damage.
They also measured systemic inflammation.
Oh.
Because often, during a severe infection, it's not actually the virus that kills the patient. It's the body's own hyperinflammatory response, a cytokine storm that causes fatal tissue damage.
Right, the immune system basically overreacts and destroys the house trying to kill the spider.
Exactly. But the TVA pups showed significantly lower levels of harmful systemic inflammatory cytokines, like IL-1B, in their lungs. Their immune response was not just stronger, it was much more precise. They cleared the pathogen without destroying their own tissue in the process.
Okay, let me play devil's advocate for a second, because I know you listening might be wondering this too.
Go for it.
If a mother is eating a different diet, like the high TVA diet, maybe she's just producing better antibodies in her milk. How do we know this survival benefit in the baby mice is actually coming from their newly folded T cells and not just mom passing down her own immune protection?
That is exactly the kind of rigorous skepticism the researchers applied, and their solution was brilliant. They performed what's called an adoptive transfer experiment. It's the gold standard in immunology to prove exactly which cell is doing the work.
How does that work?
They took just the naive CD4 positive T cells from the pups who drank the TVA milk. They completely removed the mother, the breast milk, the antibodies, everything else. They isolated only those reprogrammed T cells.
Wow, okay.
Then they injected those specific cells into regular, unexposed recipient mice who had never seen TVA in their lives.
And then they infected those recipient mice with the flu and salmonella.
Yes, and those totally naive recipient mice suddenly gained the enhanced infection protection. It was definitively the reprogrammed T cells doing the work. The superpower was locked permanently inside the genetic folding of those specific cells, entirely independent of the mother's antibodies or the breast milk itself.
That is just incredible. Okay, so we've mapped the entire chain reaction from a molecule in dairy to defeating the flu in mice. But, it's a running joke in science that we've cured mice of everything 1,000 times over.
That's very true.
The real question is, does this apply to human babies? Are we just looking at mouse biology?
Translating to humans is always the biggest hurdle. Fortunately, the researchers moved directly into human clinical data. They looked at a cohort of human mothers and their preterm infants in the Neonatal Intensive Care Unit, or NICU. Preterm infants are particularly vulnerable to infections and immune dysregulation because their immune systems haven't had the full 9 months to develop. So this is a critical, highly fragile population to study.
And they found a direct correlation, didn't they?
They did. First, they confirmed the delivery mechanism actually works in humans. High levels of TVA in the human mother's breast milk perfectly correlated with high levels of TVA in her preterm infant's blood. The transfer happens identically to the mice.
But did it trigger the same immune shift? That's the million-dollar question.
It did. When they analyzed the blood of those human infants, high TVA correlated with high levels of IL-2, which is a key cytokine marker for T-cell expansion. And even more importantly, it correlated with a high ratio of interferon gamma to IL-4.
Let me guess. That ratio is the immunological signature of the Th1 SWAT team shift we talked about earlier.
Exactly. It proved the human infants were undergoing the exact same Th1 immune polarization seen in the mice. It wasn't just a mouse phenomena.
But what I love about this paper is they didn't just stop at blood markers. They found a tangible clinical outcome tied to this in the NICU. Higher breast milk TVA was associated with a significantly reduced incidence of bronchopulmonary dysplasia, or BPD. For those who might not be familiar, what exactly is BPD?
BPD is a severe, sometimes chronic lung disease that affects premature babies. Because their lungs are underdeveloped, they often need mechanical ventilation and extra oxygen to survive. But this can cause severe inflammation and scarring in the fragile lung tissue.
So the fact that higher TVA in the breast milk correlated with less BPD means this isn't just an abstract shift in blood markers, it is correlating with actual real-world respiratory protection in the most vulnerable human babies.
And to absolutely seal the deal mechanically in humans, the researchers did one more in vitro test. They took human-naive CD4 positive T cells directly from umbilical cord blood, put them in a petri dish, and treated them directly with TVA. It caused the exact same Th1 polarization via the exact same CTCF origami molecular pathway. The mechanism is identical in humans.
So what does this all mean? How should a listener apply this to how they view infant nutrition? Because it fundamentally shifts the perspective from just counting calories to seeing food as cellular communication. But there's a catch, right? Like a very specific time frame where this software update actually works.
If we connect this to the bigger picture of human development, we have to talk about the concept of early life immune imprinting. And yes, timing is everything.
The researchers wanted to know if this was just a temporary boost, like drinking an espresso for a quick burst of energy while the baby was actively drinking the milk, or if it permanently changed them.
Exactly. So they took mice and gave them the TVA-enriched breast milk only during the 3-week nursing window right after birth. After weaning, those mice were put on a completely normal non-TVA diet for weeks. By the time they were young adults, at 6 to 10 weeks old, the TVA was completely gone from their bloodstream.
But the upgrade remained.
When those adult mice were challenged with the influenza virus weeks later, they still had enhanced survival rates compared to the control group. Their T cells were still primed. The dietary instruction given during that tiny window of infancy had permanently set their lifelong immunological baseline.
But here's the crazy part. If the researchers waited and fed normal mice a TVA-enriched diet later in life, say during weeks 5 to 8, completely missing infancy, the imprinting did not happen. They got no lasting protection.
This proves that there is a narrow, critical developmental window. Right after birth, the immune system is highly plastic. It's essentially sitting there, completely malleable, waiting for environmental and dietary instructions to tell it what kind of world it has been born into.
Wow.
Once that window closes, the immune system sets like concrete. The same molecule no longer has that permanent foundational epigenetic effect.
Okay, we have covered a massive amount of ground today, from the macro down to the molecular. Let's do one final recap of this astonishing journey for you listening.
Good idea. Wrap it all up.
We started with a single dietary fat, TVA, found naturally in ruminant foods like beef and dairy. A mother consumes it, and it passes into her breast milk. In the baby, it acts as a molecular key, releasing the GPR43 parking brake on the immune cells.
Taking the brake off.
That triggers an internal cellular alarm, sending messengers to tell the origami master, CTCF, to reorganize the folding of the DNA. This reorganization builds a robust, primed SWAT team of naive T cells perfectly calibrated for a Type 1 defense against viruses and bacteria.
A permanent SWAT team.
Exactly. It protects the infant from pathogens in the short term, heavily reduces the risk of lung disease in preemies, and because it happens during a critical early life window, it leaves a permanent, lifelong upgrade on their immune system.
It really is a stunning piece of science. And it highlights that food is a biological language. This research could eventually revolutionize dietary guidelines for expecting and nursing mothers. More urgently, it might lead to entirely new formulations of infant formula.
Which is huge.
Yeah, right now, formulas are designed around broad macronutrients, getting the fat and protein ratios right. But they lack these specific, highly targeted signaling molecules like TVA. We are realizing we've been missing parts of the software code.
It's just wild to think about. It makes you look at a simple glass of milk or a piece of cheese completely differently. It's not just calcium and calories, it's instructions.
It really is.
Yeah.
And I want to leave you, the listener, with a final provocative thought to mull over or explore on your own.
Let's hear it.
If a single, naturally occurring trans fat like TVA has this hyperspecific evolutionary mechanism to jumpstart a baby's immune system, what other single isolated molecules in our ancestral diets might be acting as hidden master switches for our biology?
Oh, wow.
We spent the last 50 years terrified of all trans fats, nearly throwing the baby out with the bathwater, literally. What other switches have we completely overlooked in our modern obsession with just counting calories and avoiding broad categories of food?
That is a phenomenal question. It really brings us back to that software update we talked about at the very beginning. If the human body is the hardware, we might just be beginning to learn the language of the code we've been feeding it for millennia. Thanks for joining us on this deep dive.